has a subtitle Search for Antiviral Drugs Against SARS-CoV-2 Using Machine Learning Methods. Our main goal was to develop a machine learning model that allowed us to screen small molecules and identify which could have higher antiviral activity against the SARS-CoV-2. To carry out our analysis, firstly, we made an homology and phylogeny analysis of the 3CL-Pro protein. We found that the 3CL-Pro is a promising target to develop antiviral drugs against the SARS-CoV-2. This protein is codified in the polyproteins 1A and 1B of the SARS-CoV viruses, getting different nomenclatures. Later, we obtained databases for small molecules. All the molecules selected had a half maximal inhibitory concentration activity, or IC50, which was previously determined by in vitro assays against coronavirus species. Here, the small molecules that were under the IC50 threshold were labeled as actives, while those that were over the threshold were labeled as inactives. The descriptors were obtained following a CDK graphic implementation in Java and using the package CDK in R for each database of small molecules. After the cleaning of data, we construct an optimized predictive model by ML for searching antiviral drugs against the SARS-CoV-2. Here, we choose the best model by testing several parameters, such as the ROC curve, accuracy, sensitivity, and specificity, to finally obtain an optimized model. And we tested three different models, the decision tree, random forest, and naive Bayes, and modified them until they fitted our data. Then we determined which of them had the highest performance and best prediction of potential antivirals. In the results, after the similarity analysis of the 3CL protein sequences of the viruses, we obtained the following. As you can see in the figure 4a, in the evolutionary analysis, we found that the coronavirus species analyzed here are evolutionarily very close, showing a 97% probability of being grouped. And in the figure 4b, we display the high similarity between the SARS-CoV-1 and 2, where the p-values of distances among individuals of each group were equal to 0, while between groups it was of 0.04. Later, we obtained the descriptors and developed several databases. In the table 3, we summarize the numbers of assays and molecules obtained with an activity against the SARS-CoV-1 and 2. And in the table 4, we show the 8 databases obtained in this study, which were vastly varied in the number of molecules and descriptors. After performing the analysis of our models, we obtained 8 databases of small molecules with the descriptors with an IC50 activity against the SARS-CoV-1 and 2, and the machine learning models that better classified the active or inactive molecules were random forest and decision tree. As conclusions, we deduce that the most important factor when creating databases depends more on the number of descriptors obtained, their quality and lower correlation than the approach used to get them, and that using these models it will be possible to find molecules with a biological activity for being used as potential treatments against the SARS coronavirus species. As perspectives, we will explore several settings for each ML model and modifications in the data composition by selecting different descriptors or joining up some of the 8 databases already obtained. And we will analyze new sets of small molecules applying the most optimized models to screen candidates that could be experimentally analyzed. Finally, we would like to thank the Universidad Oberta de Catalunya for its technical and academic support, and thanks for your attention.